Ngalakin kadijan nyunga boja.
The topic of this lecture is the phosphoprotein phosphatases, otherwise known as protein phosphatases. We're firstly going to look at a general overview of the phosphoprotein phosphatases and look at the two general groups of phosphatases, serine-threonine or tyrosine phosphatases. We'll finish this lecture by looking at pseudophosphates and the role of phosphatases in disease. Just like with protein kinases in the last lecture, phosphatases are roughly divided into two classes, and this is dependent on the phosphoamino acid whose hydrolysis they catalyze. For serine-threonine phosphatases, these catalyze the hydrolysis of a phosphoserine or phosphothreonine amino acid in proteins. For the tyrosine phosphatases, they catalyze the hydrolysis of phosphotyrosine in proteins. There are also some phosphatases that have dual specificity. They catalyze the hydrolysis of both phosphoserine or phosphothreonine and also phosphotyrosines in proteins. Now, it's important to note that phosphoprotein phosphatases are not just off switches. Their role isn't just to shut down the signaling that was activated by protein kinase phosphorylation events. They are capable of being regulated signaling molecules in their own right, and we will see that they can be involved in many disease states, including cancer. Firstly, we'll have a look at the phosphoprotein phosphatases that are serine and threonine specific. These can be broken into three families. The first family are the phosphoprotein phosphatases, or PPPs. These are shown in the top panel. The PPP family have generally well-conserved catalytic core domains, with conserved residues that are involved in metal ion binding. These are shown in red. And conserved residues that are involved in phosphoryl group binding, shown in blue. These phosphatases do bind to metal ions, such as zinc or iron, in the case of PP2B, even though they are not classified as metal-dependent protein phosphatases, like PP2C. These metal ions are thought to bind water and activate so that it neutrophilically attacks the phosphorus of the phosphoryl group of a phosphoserine or phosphothreonine residue. The second family are metal-independent protein phosphatases, or PPMs. This is shown in the middle panel. The metal ions, usually magnesium, bind to the phosphoryl group on the substrate. Water then neutrophilically attacks the phosphoryl group phosphorus, which is assisted by D or E residues acting as a base catalyst. The third family are aspartate-based phosphatases, such as FCP, transcription factor IIF, associating component of the RNA polymerase II carboxy terminal domain phosphatase, and SCP, or small carboxy terminal domain phosphatase. These enzymes use an aspartate residue for catalysis and carry the active site signature DX, DXT, or V, which is shown here. These enzymes dephosphorylate the CTD of the largest subunit of the RNA polymerase II. Let's now take a look at some specific examples. Firstly, PP1, or protein phosphatase 1. PP1 is composed of two main subunits, a catalytic subunit and a regulatory subunit. The catalytic subunit has no specificity for a particular protein target. However, there are over 200 different regulatory subunits, and it is the regulatory subunits of PP1 that confers its substrate specificity. This is in terms of which phosphoprotein the PP1 will target by providing the binding sites for these proteins. PP1 plays an instrumental role in the glycogen metabolism through its responsibility for interconversion between phosphorylase A and phosphorylase B, switching between relaxed states and T states, and on or really on and off or really off states. And this is through its active site and phosphorylation of its subunits. The PP1 enzymes can also be inhibited by specific inhibitor proteins such as I1 or inhibitor 1. If we take a closer look at the structure of protein phosphatase 1, we can start to understand its mechanism. In the top panel, we can see the structure of the catalytic subunit, shown in blue, of PP1. PP1 is bound to an okadaic acid, or OA, shown as the yellow ball and stick molecule, and we can see a distinctive Y-shaped surface groove, shown in pink. And this is defined by the three domains of PP1. We can also see two metal ions, which are shown as red spheres, bound to the catalytic subunit of PP1. These are manganese and iron. In the second panel, we can start to understand the conserved residues of the PPP family. In PP1, there are metal binding amino acids, and these are 6 highly conserved amino acids across the PPP family. We can see in the leftmost panel 2 metal ions being bound by these 6 conserved residues. On the right, this shows within the family of PPP, those 6 residues are highly conserved. So the catalytic binding site of the phosphoprotein phosphatase is crucially important, and this is why these residues are highly conserved across the family. We can take a look at the action of PP1 in this example. When protein kinase A is activated by an increase in cyclic AMP concentrations, the protein phosphatase 1 is inactivated. And this is through the activation of inhibitor 1 or I1 by its phosphorylation. Protein kinase A, when active, will phosphorylate inhibitor 1. This will then combine with our active protein phosphatase 1 to reduce its activity. By doing so, protein phosphorylation is amplified. Later, when the cyclic AMP signal is switched off, the protein kinase A will become inactivated, and the inhibitor 1, which has been phosphorylated, will be dephosphorylated. This then activates protein phosphatase 1, which can go on to dephosphorylate the substrates of protein kinase A, which includes any of those kinases that have been activated by pKa. Also noteworthy is that the source of the phosphoryl group is from magnesium ATP, which is used by protein kinase A to phosphorylate inhibitor 1. Our second example is PP2B, or calciurin. The PP2B enzyme is a heterodimer. It's composed of a catalytic subunit and a regulatory subunit. The catalytic subunit is known as CNA, or calciurin A. In the diagram, this is shown in blue. The regulatory subunit is known as CNB, or calciurin B, and this is shown in gold in the diagram. The catalytic subunit can bind to metal ions such as zinc or iron, which are shown as red spheres, in its active site. The regulatory subunit can also bind to calcium ions in its binding site, shown as green spheres. This CNB has a binding helix which also interacts with two calmodulin or CAM-like domains. We see that the activation of the PP2B requires calcium binding to the CNB, the regulatory subunit, and also calcium calmodulin binding to the CNA for its activity. We can have a look at an example how PP2B is involved in signaling systems. In a signaling system that produces an increase in the intracellular calcium concentrations, this could be through activation of phospholipase C, which causes release of inositol triphosphate from the PT-INS 4,5 biphosphate. This would induce the release of calcium from the endoplasmic reticulum, and also we would see an influx of extracellular calcium into the cell. This increase in the intracellular concentration of calcium will activate our PP2B enzyme. An example of this is the activation of transcription factors involved in T lymphocyte activation. PP2B is an important positive activating component of T lymphocyte activation. We see that when a T lymphocyte receptor is activated, this will lead to an increase in the intracellular concentration of calcium, which then activates the PP2B enzyme. The activated PP2B binds to and dephosphorylates NFAT, which is the nuclear factor for activated T cells, a specific transcription factor. When dephosphorylated, NFAT is promoted to translocate from the cytoplasm into the nucleus. This is where it can bind to DNA that encodes cytokine genes and stimulate their transcription. Another mechanism which we can look at is the control of PP2B activity, which involves the presence of calcium and also modularity calciurin inhibitory proteins. The MCIP acts as a competitive inhibitor with respect to the substrates of PP2B. In the presence of high calcium concentrations, we see that the calmodulin or CAM combines with the phosphatase heterodimer, and so the PP2B is active. The MCIP can then bind to the CNA subunit as part of deactivating the phosphatase. It is proposed that when the NFAT levels, the transcription factor NFAT levels, exceed a threshold concentration, this then stimulates the expression of the NCIP, and so provides a feedback inhibition of our PP2B enzyme. When the calcium levels start to fall, the signaling is stopped. The calmodulin dissociates from the CNA subunit of our PP2B, and also the MCIP dissociates. This goes on to be degraded by proteolysis. And so our calciurin, or the PP2B, can become active again. Another phosphatase we will take a closer look at is PP2A. PP2A is a holoenzyme complex, and they are heterotrimers, which consist of a core scaffold subunit, shown as A. This binds to a catalytic subunit, C, and also one of several different types of regulatory subunits. Each regulatory unit type also has subtypes, which we can see here in the diagram. The PP2A is able to bind to metal ions, either magnesium or manganese. And again, the B regulatory subunits provide the enzyme targeting towards a specific substrate. If we take a look at the B quotation mark or double apostrophe group, the PR59 regulatory subunit is for intracellular localization, and this targets to the nuclear site. For other B subunits in this group, they have functions in regulating the phosphatase activity, such as through the calcium calmodulin binding. We also see for PP2A phosphatases that they are regulated by post-transcriptional modifications. The two main modifications are through phosphorylation or methylation. In the case of phosphorylation, tyrosine 307 or 307 is phosphorylated. When this residue is phosphorylated, it prevents the binding of the B regulatory subunit, and so reduces the activity of the phosphatase. In the case of methylation, where the lysine 309 is methylated, this enhances the B subunit binding, increasing the specificity and activity of this enzyme. We're now going to take a look at the signaling pathways for cell growth, survival, and proliferation, and how PP2A is involved. We can see that the two post-transcriptional modifications that occur to PP2A, methylation here and phosphorylation, are involved in its control. There is a conserved sequence, TPDYFL, in the PP2A, which undergoes the phosphorylation and methylation post-translational modifications. Where we have phosphorylation of the tyrosine 307, and this occurs by receptor tyrosine kinases, this will decrease the PP2A's activity. So we have an inhibitory signal. Where there is methylation, so a methyl group is added to the lysine 309 by LCMT1, we have an enhanced specific binding of the AC dimer to the distinct B regulatory subunit, and this provides increased enzymatic activity. The activity of PP2A is also essential in other cellular pathways. We see that phosphorylation of threonine 308 and also serine 473 leads to activation of ACT, or protein kinase B. This is associated with the activation of PP2A. PP2A is also playing an important role in the WNT signaling pathway, and this is through phosphorylation of B56, the regulatory unit B56, in the activation of the WNT pathway. We can also see that there is the pathway involving apoptosis. What we see occurring is that BAD is a pro-apoptotic molecule, and BCL2 can be both regulated by the PP2A. BCL2 has an anti-apoptotic activity. Phosphorylation of BAD suppresses it, and dephosphorylation by 2,2-PA will promote its pro-apoptotic activity. Where we have phosphorylation of BCL2, it becomes active, and its dephosphorylation facilitated by 2,2-PP2A suppresses its apoptotic activity. We can also see the involvement of PP2A in disease states such as cancer, and this involves the inhibition of a molecule, or the inhibition of PP2A by CIP2A. Where there is increased activity of the PP2A phosphatase, this is linked to reduced malignancy cancers and also enhanced therapeutic responses of those cancers. Where there is reduced activity of this phosphatase, there is a link to increased malignancy and tumor progression. What is occurring is that there is a decrease in the PP2A activity, which is enhanced by the expression of a PP2A inhibitor protein, known as CIP2A. This inhibitor of the phosphatase will prevent the inactivation of a transcription factor, MYC, which is normally dephosphorylated by PP2A. The prevention of its inactivation will lead to enhanced cell proliferation. The second cause is that inhibition of PP2A will prevent inactivation of ACT, or protein kinase B, and also DAP, both again dephosphorylated by pp2A. This then leads to enhanced cell survival and a reduction in apoptosis. The transcription factor E2F1 is a central player involved in cell cycle progression, and this is also involved in pp2A's activity in relation to cancer. The E2F1 can mediate both cell cycle progression or proliferation and also apoptosis of the cell, and it is dependent on the level of expression of its post-translational modifications, including phosphorylation. An active pp2A phosphatase can dephosphorylate the E2F1 transcription factor and prevent its stimulation of the cell cycle to progress, and also enhance its pro-apoptotic activity. If we have a look at this schematic showing the involvement of both the inhibitor, CIP2A, of our phosphatase pp2A, and also the transcription factor E2F1, the MYC transcription factor, and also the kinases protein kinase B or ACT and DAPK. We can see that in cancer, the transcription factor and kinases remain phosphorylated. And this is because the inhibitory effect of CIP2A is affecting the dephosphorylating function of pp2A. What results is the tumorigenic process, where there is cellular proliferation, the survival of cancerous cells, the ability to self-renew, and the evasion of senescence or apoptosis. And we see that this inhibitory molecule is expressed in between 40 to 90% of cancer patients. If we look at cancers that are negative for expression of this pp2A inhibitor, or where therapeutic inhibition of CIP2A has occurred, there is decreased cellular malignancy and also enhanced therapeutic response. And this is because the pp2A phosphatase is able to perform its normal dephosphorylating activities. The next type of phosphoprotein phosphatases we will be discussing are the tyrosine phosphatases. Many tyrosine phosphatases are transmembrane receptor-like proteins, which appear to be involved in cell-to-cell interactions. And these are potentially signaling receptors. These receptors have their intracellular phosphatase domains activated on receiving and recognizing a signal by their extracellular receptor component. There are other tyrosine phosphatases that are soluble proteins found in the cytoplasm. What we see for these phosphatases is that they have two catalytic domains, one of which is inactive. So for all but class 3, they have duplicated catalytic domains D1 and D2. And each subclass of the tyrosine phosphatases have a specific or unique extracellular receptor domain structure. If we take a closer look at the structure of the major tyrosine phosphatases, they all share a similar structure and mechanism in their catalytic domain. The important motifs of this domain are M8 to 10, which define the catalytic core. where motif M9 contains the essential catalytic cysteine residue and the phosphoryl binding arginine residue. Motif 8, or M8, contains the aspartate residue that acts as an acid-base catalyst. And we'll have a look at the mechanism on the next slide. In addition to these motifs, M1 provides a binding site for the phosphotyrosine and gives the phosphatase its specificity for a specific phosphotyrosine on a protein. In the mechanism, what we see occurring is the cysteine neurophilically attacks the phosphorus of the phosphotyrosine. The aspartate acts as a general acid catalyst to proteinate the leaving tyrosine product. This then results in the formation of a phosphocysteine enzyme intermediate. This intermediate is hydrolyzed by a water molecule positioned by glycine in the M10 motif, with the aspartate then acting as a general base. So a closer look at the mechanism of a tyrosine phosphatase. It's a two-step catalytic mechanism beginning in this diagram here. We can see that the phosphate group from the phosphotyrosine substrate is coordinated by a main chain of amides. And this is in the protein tyrosine phosphatase loop. There is also a side chain arginine 221, that's involved. The side chain of the active cysteine 215 is located beneath the PTP loop and has close proximity to the main chain amides. We then have the active site cysteine 215, which exists as a phthalate ion in physiological conditions, attacks the phosphorus atom of the phosphotyrosol residue. At the same time, the aspartic acid is ideally positioned to act as a general acid to donate protons to the phenylytic oxygen of the substrate, up the aspartic here. The consequence is the formation of a phosphocysteine intermediate. So this is the intermediate down here, phosphocysteine intermediate. This intermediate is then resolved by activated water, aided by the aspartate and an adjacent glutamine residue. There are also some different ways that the protein tyrosine phosphatase enzymes can be regulated: by cleavage, oxidation and dimerization, or by phosphorylation. The first, by cleavage, is where there is extracellular shedding of the domains on the outside of the membrane, and these are then released from the phosphatase. Here is an example that's shown for LA, where there's no longer able to be signaling received. The second is by oxidation and dimerization. For some receptor protein tyrosine phosphatases, or RPTPs, which includes PTP, we can see that these are present as dimers in the membrane. Inactivation of these dimers occurs through oxidation by peroxide, which has been released from growth factor receptor activation, causes the catalytic sites to change conformation and so become inactivated. Now, this type of oxidation is reversible. The last type of regulation is by phosphorylation. Here we can see that protein tyrosine phosphatase can be phosphorylated by a protein kinase, C, which will cause the detachment of the GRB2. The PTP can then attach to the PP60 SRC through the SH2 domain. The binding of this SH2 domain will then expose the phosphotyrosine at 227 in the PP60 SRC, which is given by the yellow star, which can then be dephosphorylated by the PTP, the phosphatase. Lastly, we'll discuss some pseudophosphatases. So just like pseudokinases, there are the presence of pseudophosphatases, and these are phosphatases which can maintain their three-dimensional fold and ability to bind to phosphorylated proteins. However, they do not display the ability to dephosphorylate said phosphorylated proteins in cellular processes. However, it is believed that these pseudophosphatases can serve as modulators, competitors, spatial anchors, or integrators. Modulators have the ability to modulate the activity of kinases or phosphatases by serving as dimerization partners or cofactors. They can inhibit or accelerate the kinase or phosphatase activity. As a competitor, these pseudophosphatases can bind to substrates and therefore competitively prevent access to other kinases, phosphatases, or signaling molecules. As a spatial anchor, these pseudophosphatases it is likely that they exhibit a restricted subcellular localization pattern and possibly serve to anchor as a trap a particular substrate in that location. So we can see in this example, the pseudophosphatase is anchoring or localizing the substrate inside the nucleus. And lastly, they can act as signal integrators. So it is probable that in proteins with a modular composition of domains, that we have the pseudophosphatase that brings the different signaling components together in the cascade, and that will then result in a biological effect. We can take a look at an example of protein tyrosine phosphatases in disease. So the PTP1B has been shown to have a prominent role in metabolic signaling, particularly in obesity and type 2 diabetes. And this is through its interaction with IR and the JAK2 downstream of the leptin receptor. The PTP1B is also been shown to have pro-tumorigenic properties in both breast and prostate cancer when it interacts with the P62 dock. There is also a significant amount of evidence starting to be gathered that links the PTP1B to ER stress through its interaction with PERK. There's also evidence that the PTP1B can influence inflammation through other various JAKs and STATs. And it's also been indicated to be involved in heart disease. And finally, let's put it all in perspective. The phosphoprotein phosphatase 2A accounts for the majority of phosphoserine and phosphothreonine dephosphorylation in eukaryotic cells. And we know that it contains regulatory B subunits that help to target it. However, there is a limited number of PP2A regulated phosphorylation sites that are known. Now, this hampers our understanding of the site-specific dephosphorylation and also how it can suppress tumours. We know that the PP2A can selectively dephosphorylate individual sites, but how that occurs, we still do not know. It is believed that there is some kind of key and lock model for how the PP2A active site selects some phosphorylation sites and not others. This particular study by Cruz focused on PP2A B56, which is known to be a major tumour suppressor. They looked at the global substrate identification of these hollow enzymes as a strategy to identifying these interactions. What they found was that the B subunits directly affect the dephosphorylation site preference of the PP2A catalytic subunit. Further patterns indicate affinity and position are coordinated by the B56 binding motifs. It was found that specific sites with a proline in position +1 reduce dephosphorylation of phosphorylation sites. And it was found that screens to identify phosphorylation sites in the cancer target ADAM17, an essential transmembrane metalloproteinase, are regulated through this conserved B56 binding site. By binding of PP2A B56 to the ADAM17 protease, there is a decrease in growth factor signaling and also tumour development in mice.